Okay.
Okay, I can start the recording. Um,
Okay.
Hi everyone. Thank you so much for coming. Zhuoqi is currently a PhD student at Shanghai Jiaotong University, majoring in computational structural biology. His research interest mainly focuses on protein design with deep learning, with broader interest in protein conformation generation and molecular dynamics simulation. And today, we're very excited to have him present Motif-bench. So feel free to get started whenever you're ready.
Okay. So thanks for the invitation and the introduction, Divya. So today I'm happy to share our recent work on the motif scaffolding benchmark. So let's begin. Now, before we enter in the story of Motif Bench, first I'm going to let you take a look at the outline today. So first, we're going to make some brief introduction on both the design of protein and also the motif scaffolding problem. And then we're going to have a look on the recent developments of the motif scaffolding methods. And here we have the key question is that why do we need such a unified evaluation suite for that? And also, then we're going to go through Motif-bench and finally a GitHub tour on how to evaluate your own methods in a minute. So let's get started now. So we all know that proteins are powerful machines that perform numerous important roles in the biological functions and cellular processes. So proteins are contained with the primary amino acid sequence, which can fold into a relatively stable three-dimensional structure. And then the arrangement and the dynamic properties of the structures are closely related to the functions of the proteins. So here, in the modern protein design pipeline, we often have going to define a desired property, and then we used some methods, either based on the energy functions or deep learning methods to generate an ideal structures that can hold these desired properties. So this is what we call the function to structure regime. And also you can, of course, use with the aim of protein language models to directly generate sequences with the desired properties. But we're going to focus on the first type of situation today. So now that we have already generated a structure, it is not enough because the structure might be only have a blueprint or the overall topology, which we often call a scaffold, and it only contains like about some backbone atoms, or in some cases, only the carbon alpha atoms. So in this sense, we need to find an optimal sequence compatible with the scaffold that can hold this structure, also with the desired properties. So this is what we call the fixed backbone sequence design. So how do we know if the sequence really compatible with the structure? It is that we're going to use the advancement of the structure prediction methods like AlphaFold series. We're going to predict the sequences and use the predicted structure to compare with the similarity of the with the originally generated scaffold. So here lies on the key concepts of the evaluation on the protein backbone generation methods that we often referred as the self-consistency, which is to test whether the designed scaffold are really compatible with any of the sequence it could find. So we have already know some concepts of the protein backbone generation. So now we're going to take a look at the motif scaffolding. So although we know that proteins performs various functions, and sometimes these functions are governed by a subset of residues, which is often we referred as motifs. So the motifs could spread across a wide range of scenarios, such as in the enzyme active sites, the spatial arrangement and orientation of the side chain conformation of the specific residues could play key important roles in the enzyme catalytic functions. And also in some cases, like we have surface fingerprints for protein-protein interactions, and also the binding hotspot residues for the binders. So after we have the identification of the motif, either through some literature research or experimental means or some computational prediction algorithms, we're going to use some methods to design a scaffold around this motif, which is often we call motif scaffolding. So the aim of motif scaffolding is to design diverse scaffolds while already maintain the original biological functions. So how can we use motif scaffolding to change our lives? So here we have some examples, like this is a work that about 10 years ago that where the scientists first identify the epitope of an RSV protein, and then they use iteratively ab initio folding and design with some optimization to gain a final protein that can mimic the epitope of the virus protein and then to induce the antibodies. So in this sense, it has great potentials in the vaccine design. And here in another example, we're going to see how scientists use motif scaffolding regime to design binders, which have some high binding affinity to selective proteins. So here they tried two strategies. Like the first one is the identification of the hotspot residues on the interface of two proteins. And then they either use the motif grafting strategy that transparent the hotspot residues onto some existing scaffolds, or directly de novo assemble the binder, which can then selectively bind and kill some infectious cells to reduce the production of virus. So the two methods I introduced here are some pre-deep learning stages, which how they do motif scaffolding. So what about the recent years with the advancements of these learning methods? So basically, there are many schemes for designing motif scaffolding proteins in this sense, and I'll select two of them with some two early works representing both categories. So the first one is what we call hallucination. Generally speaking, it is used to a random sequence and then use a structural prediction module to get the structure of the random sequence, and then use some loss functions upon our desired properties. For example, in the context of motif scaffolding, it might be the geometric similarity between the desired motif and the motif in the initial sequence. And then we can do some gradient updates or sampling techniques to iteratively get a sequence closer to what we call a novel scaffold containing the motifs. And another type of methods is based on generative models, which either trained on the score matching objective or diffusion objective, the score matching objectives or the flow matching objectives, as many of them are something like the VAEs or diffusion models, which they aim to first learn the distribution of ideal protein structures, and then they treat the motif scaffolding problem as a conditional generative problem where the condition is the motif and the object is to design an ideal protein structure that can hold the motifs. So both of these methods have already produced some successful motif scaffolded proteins, and many of them are already being experimentally validated. And most of all, with the advancement of deep learning techniques, we can get a huge bunch of the candidates for to be selected into the downstream application and wet lab experiments. So, with such an avalanche in the motif scaffolding methods, so we certainly have an interest of how this method performs, like which of them performs like relatively the best, or how different methods performs in different motifs, because we have observed that many of them have very different performances between different motifs. So this relies on a convenient implementation with a suite of standardized criteria for evaluation and also a data set with various motifs to test and rank the capability of each methods. So how about the status of the evaluation on motif scaffolding? So it might seem very easy because we have already introduced how we evaluate the protein backbone generation methods to date. But when we really come to the evaluation, we're going to face some very different scenarios. So like if we want to measure the designability, we have RMSD and TM score, which both can measure the similarity between the generated scaffold and the structural predicted scaffold. And also, when doing the designability test, we're going to use the structural prediction methods. So which one do we want to use? ESN fold, AlphaFold 2, Omega fold, or some latest methods that contains the information of different types of macromolecules, such as AlphaFold 3 and its open source implementations. And when calculating the diversity, such as the pairwise TM score, and so the diversity aims to measure the differences between the generated scaffolds, so we can measure them by the pairwise TM score and also foci cluster. And also for the fixed backbone design method, because within the motif, a natural thought would be to fix all the types of amino acids the same as the reference one. But in some cases, the side chain of each motif residue might not be totally dependent on the exact amino acid types, sometimes with only a subset of side chains, or sometimes only with the backbone. So in this case, scientists also trying to relax them. And also we have many other variables, such as whether to integrate the confidence matrix in the structural prediction methods and whether to choose their threshold, and the contigs, which represent the placement of motifs. So given so many different matrix and the evaluation pipeline, we have entered a chaotic status that it's hard for us to see whether each method could perform well on different motifs. So there have already been some benchmarks which aims to solve this problem. Like a previous work by us, Scaffold Lab aims to evaluate protein backbone generation methods, both in the sense of unconditional and motif scaffolding methods. And also we have MotifBench, which covers a wide range of scenarios, like in the protein generative models, such as sequence design, structure design, and the structure conformational prediction. And also we have shapes, which focus on to measure the difference of the distributions in the topology space between the designed scaffolds and the native ones. So although we have we have seen so many significant efforts in the unified evaluation of the benchmark, they have already there are still many discrepancies between the real matrix and also between the different matrix and the pipeline. And also a major limitation is that when coming to the problem of motif scaffolding, most of these methods measure them with a test cases set brought by RF diffusion, containing about 25 cases. And to date, most of the state-of-the-art methods are able to solve more than 20 of them, which means that what we see today with the latest methods are mostly some marginal improvements upon previous methods. So this additionally makes us harder to compare which method works better and whether their ability are able to solve completely novel motifs. So to trying to tackle these issues, this is the motivation we mainly decide to develop MotifBench. So first, let's take a grand view of the MotifBench. It has three main components with a new set containing 30 test cases across various scenarios, such as the enzyme active sites, the interfaces, and also the orphan proteins. And we have a suite of standardized matrix and the evaluation pipeline, along with a leaderboard that record everything that each method performs. And also, we're going to have an easy-to-use implementation that make the evaluation of motif scaffolding at ease. So we're going to go through the three components sequentially. So let's first take a look at the part of the evaluation pipeline. So the first question is that how do we really specify the scaffold to be a success or not? So suppose we now have a design scaffold, and here the orange part is the motif and the gray part is the rest of the scaffold. So we're going to first use a fixed backbone design sequence method, which we picked protein PNN here for its robust performance that has been validated by many wet lab experiments. And we generate it. And we're going to generate 8 sequences per scaffold. And then we use the structure prediction method to predict all the structures of the 8 sequences, which we use ESM-fold here as its efficiency and also its competitive performances in de novo proteins. So after we get these predictions, how do we measure a scaffold is success or not? So we're going to see them in two of the perspectives. So the first one is the motif reconstruction that we aim to resolve whether the similarity between the motif and the reference motif and the generated structures motif are close to each other. So we've set the threshold as 1.0 angstrom here. And also we want to have the information that whether the generated scaffold has the ability to find a compatible sequence towards it. So here we have this self-consistency RMSD where the threshold is set to 2.0 angstrom here. So the two matrix corresponds to the motif maintenance and the scaffold validity. And for each scaffold, if any of the 8 sequences generated by protein PNN has satisfied both of these criteria, we would call the scaffold to be success. So how about the complete pipeline? Suppose we're a method developer and want to evaluate the motif scaffolding on different test cases with MotifBench. We're going to first, like for this motif, we're going to have to design 100 scaffolds. And then we're going to test whether they are success based on the aforementioned criteria. And then we have 3 main matrix to define the capability of this method. So first of all, we're going to test what we call the number of unique solutions, which is going to measure the number of clusters we detected among all the successful scaffold. So from the perspective of method development, we want to see that whether these methods have the generalizability upon different topologies, or they are only seek to find one topology that continuously to be the solution to guess. And also from the for the perspective of design, because we want these scaffolds to have very different properties that could improve the utility in the scaffold in downstream experiments, such as the expressivity and whether they are soluble and so on. So we want to have our candidates to be diverse enough to improve the success rate in our real-world experimental design. So we're going to first use FOSI cluster to cluster among the successful scaffold and count the number of cluster, which is here we call unique solutions. And also, the modern motif scaffolding methods have a major differences compared with the previous motif grafting procedure, which is that we want the methods to generate completely novel scaffolds instead of just grafting the motif onto some existing scaffolds, because this would allow a larger search space compared with the existed protein database. So here we're going to use FOSIC search to search the successful scaffolds against the PDB database, and then we're going to find the closest protein with the similarity between the design scaffold and calculate the TM score between them. And the final novelty value is 1 minus this PDB TM score, where a higher novelty value indicates that the scaffold is more novel. And finally, we have this success rate, which was simply defined by the number of successes among each of the scaffold set, where here in MotifBench, we have 100 designs. So here I want to mention some differences between our evaluation pipeline compared with most of the previous evaluation. So the first difference lies on the computation of novelty. So for the previous evaluations, most of them just sequentially calculate the novelties using closed search or other methods. And then we get a novelty value for each scaffold, and then we directly average by scaffolds. So this can indeed reflect the properties of novelty for one scaffold set. But consider we now have a scaffold set like in this case. The three of them have a very similar topology, but the rest of them are pretty different compared with them. So we'll have the final novelty value pretty biased towards this dominant topology. So in this sense, we might get a biased novelty value to evaluate the method. So how do we treat that in MotifBench? We're going to use a normalized novelty calculation by clustering. So we're also going to compute the novelty value of each successful scaffold. And the difference lies that we're going to first calculate the mean novelty value of each cluster. And then the final novelty value is the cluster value by average. So in this sense, we will get a more unbiased calculation of the novelty value to get a fairer comparison. And also, another key difference lies on the definition of contigs. So we know that many of the motifs has many different segments of them. So the contig in this context means that how do we choose the order of motifs and also the scaffold serve as intermediates. So the previous methods often follows the original contig, which brought by Artifusion, or makes some small adjustments and to let the motifs to be placed sequentially. And also, the intermediate scaffolds are often chosen from within a range randomly, so which makes that the overall length of the scaffold are different by scaffolds. So from the design perspective, like the sequential order would make that the search space to be limited. And also from the perspective of evaluation, we all know that the diversity calculation and the novelty calculation are pretty sensitive to the overall length of the scaffolds. So this would make the evaluation more stochastic. So here we make a new rule of the evaluation that the motifs could be placed interchangeably within the sequential order. And also the scaffolds could also, each part of the scaffolds could also be changed by their lengths, but the overall length of the scaffold are fixed to a certain value around its reference scaffold. So in this sense, we get a fixed overall length and more flexible motif placements that both enlarge the search space when designing these scaffolds and also make a more fair comparison between different methods. So after the introduction of the evaluation pipeline, so let's take a look at an updated set of the benchmark cases. So as we mentioned before, most of the methods evaluate their scaffolds on the Artifusion benchmark, which contain 24 monomers. And it is observed that most of the advanced case methods could solve most of them. So here we want a much more difficult and diverse benchmark set to represent different scenarios of motif scaffolding problems. So we first have 6 enzyme active sites, with one of them from the serine protease and 2 of them from de novo protein scaffolds, which developed by regional design strategy many years ago, and 3 enzyme active site with known mechanism in the database of mechanism and catalytic site atlas. So after that, we have 8 from the original RF diffusion test set, which contains some representative and problems that might have big application in the downstream applications, such as the viral epitopes. And also we have 8 motifs which we extract from the latest, often proteins that has little or no homology to existed ones, to evaluate the generalizability when the methods, how they perform in these outsiders that they haven't seen in the training set and have little or no similarity compared with existed protein structures. And we also have 4 from the ESM3 test cases. And we have 4 additional variations where the 3 of them are length varied cases. This is because we have observed that even with the same motifs, the different methods would behave very differently when coming to different lengths of the overall scaffolds. And in practical, we often want to have different usages of the scaffolds. Like in some scenarios, we might want the scaffold to be as small as enough, and others to be larger. So here we want to set 3 length varied cases that has 2 different overall scaffold lengths with the same motifs to test that whether these methods have the capability to generate longer or shorter motifs, or shorter overall scaffolds, or how they behave differently from each other. And also we have 1 segment varied cases from a binding interface which contains in total 4 segments, and we have one problem for 2 of them and the next for all of them, to see the difficulty compared by given enough or not enough situation information to recover the complete epitope of and the rest of the scaffolds. So here we have some pretty interesting cases here to share. Like the first one is was extracted from a parallel beta strand, where here you can see in the left wing of this, we have this 4-term parallel beta strand, and we extracted 2 of them. And we want the methods to find out that whether they can recover the middle of this strand, given the 2 point of them. So another pretty interesting example is the green fluorescent protein. So often represented by a kinked alpha helix that might be crucial to the formation of the chromophore, and some other residues that might play the roles in the electrostatic interactions between them, and surrounded by an 11-strand beta barrel. So previously, scientists often use mutations to design new GFPs, about up to mutating about 20 proportion of the all residues. And then in the ESM3 paper, they managed to find a solution that with at least as about 60 sequence identity to all the non-existing GFPs. So here we place this problem here with the minimal motifs, and we kept some important residues that central to its function. And also we want to ask that whether the motif scaffolding methods could redesign this GFP. And also a more promising outcome is that if there exist some methods could design a new scaffold apart from the topology of this beta strand, of this beta barrel, to explore completely novel scaffold topology. And another known problem in the motif scaffolding problem is that sometimes the number of discontinuous motif segments has a relatively strong correlation to the difficulty of the problems. So here we divide all of our problems by 3 sets. The first set here are cases containing only a single continuous segments, and the group two is for the cases that containing double segments and multiple segments, which we propose pretty challenging for these methods. So here we have another question that, given so many different motifs, how do we rank various methods in a unified way upon all these different motifs? So here we have this MotifBench score, which um, is mainly driven by the number of unique solutions with a control factor of alpha. And then we will get a MotifBench score for each of the all motifs, and then we can average across the test cases to get a final MotifBench score for one single methods. So what do we have in this MotifBench score? So first, it ranges from 0 to 100, which is a desired property that we want to illustratively see how the method performs compared with each other. And also, um,
Um, Sorry, I think we can't I can't hear you. Can others? Leo, can you hear?
hello, can you hear and see me?
Yes, all good now. Thank you.
Okay, I'm going to. So what were we when you last hear from me?
Just the last maybe 30 seconds to a minute cut out.
Okay, so I'm going to reshare the screen. So is it that we were in this slice?
Yes.
Okay, so I'm going to continue. Sorry for the interruption. So the MotifBench score is going to reward the methods that find few solutions with many of the cases, rather than to reward the methods that find only a few cases with many solutions. Because we want the motif scaffolding methods to find solutions for as many problems as they are, rather than they can only tackle a few problems with many diverse solutions. And this is done by the control factor of alpha, which controls the increase as the number of solution grows within each case. So, for example, when we have the solutions equals to 1, we have a MotifBench score of 17.5. And if we have 5 solutions, we have a score of 52.5. And then if we already have 50 solutions out of 100, we already have a score of approximately 100, which is the perfect status. So So by this kind of formulation, we ensure that the score will reward the methods that finding the first few solutions in the case rather than the marginal improvement upon pre-soluted cases. So we now have this matrix and test cases. So to test the utility of MotifBench, we first evaluate MotifBench on RFT-Fusion, which is one of the most powerful motif scaffolding methods and also validated in experiments. So the first question is that how many problems is RFT-Fusion capable to solve in MotifBench? The answer is we get 14 out of them, approximately half of the 30 problems to be solved. So this is a lot less compared with in the 25 problem dataset that in many of the reported evaluations that RF diffusion and other methods were able to solve many or even all of them. But this could owe to many different reasons, such as it could possibly be owed to the difficulty of MotifBench test cases as increase, but it could also because of some internal limitations of the evaluation pipeline. So in this sense, we throw a question that how does the MotifBench pipeline could perform with the reference scaffolds, which the motifs are extracted from? So the answer is that in the reference scaffolds, there are 20 out of 30 that could be successfully find a solution upon them. So this means that there is a clear room for improvement of the current motif scaffolding methods compared with the reference scaffolds. And another interesting fact here we got is that 2 out of the 6 cases that our diffusion failed to find, but the reference scaffolds was able to solve is 2 cases that designed by original design regime that 2 de novo enzymes, where the first one is to catalyze the camp elimination reaction, and the second one is the retroalder reactions. So this indicates that although we have seen a lot of advancements in the modern motif scaffolding methods, there are a lot to be done. There are still a lot to be done to compare with the original design upon specific cases. And thus, MotifBench the ultimate answer to the evaluation of motif scaffolding? So we certainly have very many limitations and also some efforts to be worked in future directions. So the first one is that how about to do motif scaffolding with alternative scaffold parameterizations, such as beyond the structure-based methods, we have sequence-only methods, such as evoldiff, and also sequence structure co-design methods, such as prot-prodel. So why didn't we incorporate them into in the current iteration? There has some key challenges, such as the number of evaluated design proteins should be certainly reconsidered. For example, in the scaffold scenario, we want to evaluate with protein PNN to find optimal sequences of different scaffolds of a single scaffolds. But if we have one sequence, then we cannot obviously run 8 times of structure prediction to find out whether it could behaves well on the desired motifs. And also there's not a single pipeline across all paradigms, such as for the sequence-only methods, we could consider run MotifBench pipeline under the predicted structures of the sequences. And also for the co-design methods, we could additionally compare the compatibility between their own generated sequence structure pairs, aside from the scaffold refolding pipeline. But there are still a lot to be investigated, whether we can find optimal comparison paradigm between both of them. And also there is another limitation that why didn't we encompass the sidechain confirmations into the current pipelines? This is because that we all know that the sidechain confirmations are sometimes central to the function of motifs under many biological processes, especially in the scenario like enzymatic catalytic functions. So in this sense, there are two major key challenges we got here. So the first one, the practical challenge, but obviously could be solved, is that the number of atoms are not completely the same in the motif sidechains because we sometimes have to redesign them. But this could be done by directly compare the sidechains that plays crucial functions or to first align the backbone atoms and then calculate the sidechains. But another key conceptual limitation is that the sidechains are also represented by dynamic properties like the backbones. And also when the structural prediction methods used to predict the sidechains, they are often constrained to a certain extent by the pre-organization of the scaffold themselves. So better structural prediction model incorporated with different information such as the ligand and also the cofactors and also the relaxation with the force field could certainly help with the sidechain accuracy. But this is still needed to have some further investigation. So I'm going to here to mention that many of you might be already hear that or have read the paper of After Fusion 2, which was released in about 1 or 2 weeks ago, which does a pretty good job in the context of enzyme motif scaffolding with superior performance. So the After Fusion 2 has some advanced strategies like the unindexed strategy, which largely resolves the challenges choosing right contig for the motif scaffolding problems. And also, the latest test set brought by them using the representative enzyme active site collected from the MCSA databases are great additions to MotifBench test cases, which aims to to cover more diverse scenarios of the motifs. So and also that in the After Fusion 2 paper, they also discovered that the number of discontinuous segments have a relatively strong correlation to the difficulty. So with all these advanced techniques and methods, we can certainly say that the story of motif scaffolding is far from over. So maybe that some of the problems it might seem impossible to be resolved today might be resolved by some powerful groundbreaking methods in the near future. So what would be the next iteration of MotifBench having? So we're looking forward to the first of all is the baseline results of different methods, which we're actively running and collecting. And also, as mentioned before, the integration of motif scaffolding with various paradigms, and also better matrix along with the evaluation pipeline and more collections of representative test cases. So it is worthwhile that during the process of these projects, we initially did collect more cases rather than the 30 cases here. But the evaluation process has a great computational cost to be burdened with. So to let people to run a reasonable computational time with a test set that contains various problems. So we finally cut down the test cases to be the number of 30. And since we develop MotifBench for the better reproduction and also for the ease of motif scaffolding developers, we aim to leave some community guidelines for better reproduction here, such as the recording of the scaffolds and the metadata, and along with the evaluation results and the configs that ran the evaluation. And this will all be hosted along with our leaderboard. And after all, since, as I mentioned just now, the computational burden is a lot when evaluate the motif scaffolding methods, particularly on the test of designability. So as we have already had a standardized pipeline here, we encourage the future method developers to directly cite the stats and data from the leaderboard. So without the burden to re-evaluate everything under different scenarios and evaluation matrix and pipelines. So finally, we're going to have a tour on the MotifBench GitHub repo to see how we can easily launch the motif scaffolding evaluation. So basically, the MotifBench GitHub repo contains of three main components. Like the first is the place to host 30 test cases, and the main evaluation pipeline, and also finally a leaderboard. So how do we specify the test cases here? So first, we're going to specify the motif of each problem, which in the two columns here, we will have the ID of the PDB and also the motif indices of the residues to be extracted with. And after that, we have done some little research to define the residues that play key interactions of the motifs, and then we can leave the other of the motif residues could be redesigned during the fixed backbone design procedure with protein PNN, which allow for a larger search space in the sequence design step. And we also specify the overall length of the scaffold around with the reference scaffold, and also finally a group that with the first represent the single segment motifs, and the group 2 represent the double segments, and group 3 for multiple segments of motifs. So basically, this file contains all the information for the motif test cases, and you don't need to rewrite everything here when you're going to evaluate. So this is just a case for reference. And after we have this information for motifs, we're going to have the motif information, a formatted motif PDB files, which contains the enough information for the motif scaffolding methods, both for design and evaluate. So consider the case 1BCF, which is a heme binding sites. It has 4 discontinuous segments. So here in the formatted PDB files, we have each of the segments to be represented by a different chain. Like in 1BCF, the 4 segments are represented by chain A, B, C, and D respectively. And also the segments are separated by the semicolons, and which the letters represent the motif segments, and the numbers represent the intermediate scaffold lengths of them. And the unknown residues in the placeholder of the residue types in the PDB file indicates that these residues could be redesigned during the fixed backbone design procedure. And finally, we can get a motif PDB file here that only represent the motifs to be compared with when evaluating different motif scaffolding methods. So after that, for evaluation, what you only need to prepare is 2 things. The first one is the metadata, which we call the scaffold info CSV file. And in the context of MotifBench, we have 100 scaffold to be evaluated in each case. So we have the sample number here, ranges from 0 to 99, and also the motif placements here, where the motifs could be placed interchangeably between the scaffolds. And also, when having this metadata and the corresponding motif PDB, the scaffold files, we just need to place them into the case folders, and then finally a big folder containing all the scaffolds. And then this is basically everything you need to prepare. And then you can launch the evaluation and get the results. So what we see in the outputs of the evaluation? So we have evaluation results per scaffolds, which contains in the big CSV files, and also some summarizing matrix such as the free main matrix about the success rate, the number of unique solutions, and the novelty. And sometimes in some cases, we might not have come to a solution, but we might want to see that why do our methods fail in this kind of case, and also how close I am to the solution. Because even though we didn't satisfy the criteria of MotifBench, we can also take a visual inspection of that and to see whether it could be actually used for further refinements like mutations or redesign. So For these purposes, we also have some auxiliary matrix, such as the closest contender, where we find the scaffold which is designable with the minimum motif RMSD with the reference motif. That and separately save them for the method developers could check whether their motif performs in this closest contender. And we also have some sort of visualizations on some key metrics like the RMSD of the scaffolds and motifs and the novelty, and also the visualizations of the scaffolds colored by the motifs and scaffolds separately. So after that, we're going to automatically put everything together for the problems and for the matrix like the number of solutions and so on. And we will have a summarized file for that. For each group of the 10 cases, how many problems do we solve and the main performances within each group to see how the method could be improved further based on the different kind of performances with each group. So we hope that with this rather convenient suite for the evaluation, the method developers can run their motif scaffolding evaluation at ease and focus more on the original definition, the precious time to the method development. Yes, so this is basically all the things we had today. So this is a huge team effort here. So I want to say a big thank you to my amazing collaborators, Bo, Kieran, Kevin, Jason, Joseph, and my supervisor, Haifeng, and also Brian, who kicks off and leads these projects for their invaluable inputs and continuous supports. And here we put the links here, both of the MotifBench GitHub repo and the preprint. So if you have encountered any issue when running MotifBench evaluation, or have any suggestions, comments, or some questions, please feel free to leave a GitHub issue or directly contact me or Brian. So thank you for your attention today. So that's it.
Thank you so much for your talk. I think we have one question in the chat, and then if anyone else has questions, feel free to leave them in the chat or unmute yourself. Can you see the questions in the chat, or I can read them out?
Okay, so here I found how can you assess the foldability of designed proteins and novelty of fold could be the matrix of biological functionality? Let's say protein fashion. That means the design have biological reference. Oh, that is a good point. So basically, we are not assuming that the test of the novelty directly related to the function, the actual function of the scaffold, but we rather want the assessment of diversity and novelty to be more useful when selecting potential candidates. Like for the diversity set, as I said before, we want to have some different candidates, like because the different overall topologies could bring to very different outcomes under the experimental conditions, such as whether we can express this protein or whether they are soluble, which might also be useful to its functions. And also for the novelty, and this is particularly because we want the modern motif scaffolding methods to generate more diverse and novel scaffolds compared with existing ones, because the existing ones could be found in the databases and could be directly transparent on. But I think the novelty is beyond the function and also have a sense to test whether the methods could find an optimal solution beyond the protein scaffolds they've seen before. So hope that answers the question.
Okay. Does anyone else have any questions? Feel free to just unmute. Okay, if not, I think we can end it here. Thank you so much again for speaking. It was a very interesting talk.